All right. Before we begin, does anybody have any questions over what we covered last time? I feel like I rushed through that pretty fast. But it's kind of important that you understand it so that you understand the type of analyses that they were working with when they do these regressions. Remember that they're always interested in measuring resistance by measuring baseline speed, which is the speed before they've been injected, and versus post-injection speed. And so, even though they talk about it as resistance, the resistance that they're talking about is the difference between how fast they are after they've been injected with tetrodotoxin versus how fast they were running slowly without being injected with tetrodotoxin. So even though they talk about resistance, that resistance value comes from this relationship between post-injection and baseline speed. So if you don't understand regression, there are certain things you're not going to understand about the papers that are being. No question. All right, if you have questions, you can come to either Dr. Jones or myself. We both know how to do regression. Today we're going to be revisiting quantitative genetics. You've already been exposed to quantitative genetics in the second part of the Galapagos case study. We're going to revisit that, and we're going to go into it in a little more depth today. So we've already described quantitative traits in the past, but you should be able to do that. We had a quantitative genetics projecting change in a trait over evolutionary time. Using the Breeder's equation, we're going to expand the Breeder's equation today to include more than one trait. Before we did it with just a single trait. Now we're going to do it with multiple traits. And I forgot the activity that I was going to bring today. We're going to illustrate positive and negative genetic correlations between traits, calculate the evolutionary response to selection for genetically correlated traits, and explain how genetic correlation between traits affects the evolutionary responses. Dr. Judd, I left a stack of papers. I suspect they're sitting just to the left or right of my desk where my computer screen is. Did you get those? It's a quantitative genetics exercise that we're going to work through today. So, just remember that quantitative genetics deals with quantitative traits. This is a field of study where we look at the genetics of organisms mathematically, rather than paying attention to all the individual little genes. But remember that this math is rooted in simple Mendelian processes as they apply to multiple genes, and the goal of this is to predict how selection acting on one generation affects the trait value of that population in the next generation. So, these quantitative traits are controlled by multiple genes, meaning that they're polygenic. Each dominant allele adds to the effect of the genes on the phenotype, and examples of this are ear length in corn, crop yield, fat content in meat, height in humans, eye color in humans, skin color in humans, et cetera, et cetera. So back when we covered this before, we were doing simple genetic traits, simple genetic process with Mendelian traits, and so we would start off with parental generation that is homozygous dominant for all traits, one that is homozygous recessive for all traits. When we cross these, the gametes that they can make are these, and when you combine those together during fertilization, you get heterozygotes. And so if we then allow those heterozygotes to cross with one another, and each of these dominant alleles contributes a little bit to the phenotype, what does this cross look like if you were going to do a Punnett square? Noah, what is this Punnett square coming out of this thing going to look like?
8 by 8.
It's an 8 by 8. You have 8 different gamete types that come out of this genotype of the F1 offspring. Anybody want to come up here and give us those 8 genotypes? Just to practice. Like Jackson maybe. Or Wright, for that. Right. Explain to the class as you're going.
I got the computer.
Oh, what happened? Oh, too much weight. So as you generate the gametes, explain to us how you're doing it. Whoa, what are you doing? Now do the Punnett square. Generate gametes. Generate the gametes that are going to go into the Punnett square. So remember that gametes, so these gametes here, why are these the gametes? Because we have one representative from each gene, and this is a diploid organism, meaning that it has two copies, and this is a haploid organism. Were you able to find them, or was it hopeless? Oh, good, awesome. So now this is the genotype of that. So this is the parental generation. This is the F1 generation. What types of gametes do these guys produce? Huh?
Yeah, you asking for like the big A, little a, little a, little a?
Yeah, but you have to have three genes now because you have gene A, gene B, and gene C. So gametes have to have a representative of every gene in their cross. Remember from simple Mendelian genetics. So if we were doing this cross, this guy produces little a gametes, this guy produces big A gametes. When they fertilize, you get this. If you then allow those guys to cross, this individual can produce two gamete types. This individual can produce two gamete types. And then that's what goes into your genotype. But if you do this cross, your F1 generation looks like this, but then the gametes that this individual can produce has to have an A representative and a B representative. That's one gamete that can come out. It also has to have an A and a B, an A and a B, and an A and a B. So these are all now gamete types, and they go up and down on the Punnett square. How do you make gamete types here? So I'm going to erase this so you can't cheat. Coming back to you.
Hope so.
This way of doing it is going to make my head explode.
Well,
What's an easier way to do it? A less prone to error way of doing it? Somebody help them out. How would you do it if you only had two traits? If you only had an A and B? Is there a systematic way of doing it? You guys remember? Yeah. Jonas remembers. Come help him. Stay up there, Riley. Help him only as far as he needs help, and then he'll finish it.
All right.
Go to the board.
So if, and I'm going to do this vertically because it makes everything slightly easier to digest. If we start with this, we can just start alternating that, and that can start transferring down. So after this continues alternating, it transfers down. This can alternate again, transfers down, and then we can keep doing that. And that allows us.
But you've already got a repeat in there, don't you?
I messed up the alternation.
So somebody other than Jonas, does somebody have a more systematic way of doing this? I could have sworn that we did this. No, if that works for your brain, that's fine. My brain doesn't work that way. And I don't think Miriam's does either, because she has a puzzled look on her face.
That's me all the time.
What? Oh, okay.
We called it a method.
Yeah, we gave it a name that we borrowed from math. We gave it a name, it's an acronym, 4 letters. Yes, if we want to solve this, how do we do that? First down or last, we do that, we do that, we do that, and then we do that. If we have this genotype, we can mark off the first two things here, right? And we can do what? can apply the same idea of FOIL. So we would combine alleles first, outer, inner, and last, right? What would this be? Jonas, I'm not saying your method
Yeah.
won't work. It's just that this is
I-
a systematic way
that
that
makes
won't
sense,
lead to
yeah.
mistakes.
I don't know how I messed that up.
And then now what do you have to do to get that last gene in there? Just add a big C to it and then reproduce that FOIL and then add a little C to it. Yep. And so then you get 8 different genotypes, and you know for a fact that they are all distinctive. Yeah, give Riley a point for toughing through it.
Yeah.
Once again, nothing against your method, it's just that it's necessary.
Like literally, I was looking at a Punnett square derived from that.
So,
This is what
we
it's
would-
like writing in front of people.
we would get this hideous Punnett square, wouldn't we? And we showed you how you can, in a typical Mendelian genetic sort of way, but in quantitative genetics, all that really matters is how many of the dominant allele you possess in your genotype. So we can bust that down to a pattern of number of dominant alleles. This is all review. And what does this distribution of alleles look like ultimately? What's this distribution going to look like? We have one individual in the F2 that has 6 dominant alleles. We have 1, 2, 3, 4, 5, 6 individuals that have 5 alleles. Yeah, no. It looks like a normal curve,
Yeah.
doesn't it? There's the normal curve, and this is what we see oftentimes in populations with these kinds of traits that are quantitatively based, that are based on a bunch of different genes contributing to the phenotype. Is this all coming back to you now? You recall this? Good. We gave you a way of doing this. This used to be delta Z. That's how we presented it the first time. You can easily label it R. R may be a little easier for you to remember because it's the evolutionary response to selection. You see it in the literature both ways, delta Z and R. And it's simply the heritability of a quantitative trait multiplied times the strength of selection. What's a mistake that a bunch of you made on the second test with regard to heritability when you were doing this calculation? If we had a heritability of 0.91, the strength of selection was that the trait increased 2 centimeters. When you calculated, when you tried to calculate what the evolutionary response to selection would be, what was the mistake that a number of you made on the first test? And on every one of your papers I wrote, don't do this, don't do this, don't do
I'm
this.
guessing square rooting heritability.
Yeah, so they didn't square root it. Most people who missed this, they took the value here and squared it. 8 squared, this is just a symbol. When Fisher came up with this, he wrote it as 8 squared. Remember, you might or might not remember that I told you this. He wrote this as 8 squared because this is genetic variance. And in the same way that genetic variance is a type of variance, S squared is sample variance, and so he put the square on there so that you would remember that it's a type of variance, it's a type of variation. But you don't. 8 squared is 0.91. 8 squared is not 0.91 squared. 8 squared is not 0.91 that you take the square root of. It just is what it is. Think of it as H squared equals heritability equals 0.91 in the same way that we could have called heritability Q equals heritability equals 0.91. This is just a label, a shorthand label. The squared doesn't mean anything mathematically, except it was a reminder to Fisher that it's a type of variance. And when we write sample variance, it's the standard deviation squared. And so the H should have a square on it. It doesn't really make any sense except in Fisher's mind, but that was his rationale. So if you know what the strength of selection is, if you know what heritability is, you can predict what's going to happen with a trait in the next generation. If you have this group of parents and they assortatively, they mate randomly, and there's a certain amount of selection, the next generation will be different phenotypically, and it will be different by this amount of change. So don't take the square root of it. Don't do anything but simple multiplication. The simple multiplication in this case is 0.91 times 2. And so you would expect this thing to change by 0.812 because there's pretty strong selection. Is that right? Oh, sorry, 0.182. 182 because 2 times 9 is 18, not even 1. I was squaring. So is that right? Is my math right?
That should be 1.82.
Oh, 1.82. That's it. It's only 2 decimal points. Sorry. 1.82. I'm like, that's way too little change because there's this much selection and heritability is almost 1. This should be close to 2. Yeah, so I wasn't paying attention to the number of decimals that we have up. So you should expect 2 centimeters of change in the parents to result in 1.82 centimeters of change in the next generation. And that's because heritability is really actually pretty high in this case. Remember, heritability can vary between what and what numerically? What's the low range, what's the high range of heritability?
It's 0 to 1.
0 to 1. So if you're at 0.91, that's pretty high heritability. If you're at 0.1, that's really low heritability. Do you guys remember how we measure heritability for quantitative genetic traits? How can we quantify heritability? Jackson, is your hand up? Somebody other than Jonas? Elizabeth, do you recall how to do heritability? Do you say that because you don't know or because you're unwilling to try?
No, I don't know.
Okay, call on somebody else in your group who might know. Miriam?
I'm looking, but I don't know.
Did I give you guys homework from Monday to go back and look over this stuff again? I believe I did. I believe it's the last thing I said before I let you go is go back and look over the quantitative genetics. Because this is all review, right? This was test 2 material. Point to whoever the first person is to come up with it. Is it Jonas? Now Jonas is questioning himself. Do you know it?
Yeah, no.
Yeah.
It's the slope of the regression of the graph of average offspring and average parent.
Who's on the x-axis, who's on the y-axis?
Parent is on the x-axis.
Okay, so it's the regression of the mid-parent value, the average of the 2 parents, regressed against average offspring phenotype, if the genes of the parents are 100% responsible for the phenotype of the offspring, then this will be a 1-to-1 relationship, and that will give you a heritability of 1. If the genes in the parents don't contribute 100% to the phenotype of the offspring, then this heritability will be something less than 1. Because it's the slope of this relationship. M is the slope value. It's what you multiply times the parent phenotype to translate that phenotype into offspring phenotype. And so the slope value can either be 1 or it can be 0, once again, where knowing something about mid-parent phenotype doesn't tell you anything about offspring phenotype. This would be a slope of 0, in which case there's no heritability. That trait, whatever that trait happens to be, there's no genetic basis for it. So this is trait 1 versus trait 1 in parents and offspring. Okay? We did this with height in previous iterations of this. But organisms are not made up of just a single trait. Unfortunately, things are more complicated than that. Anybody in the water bug group explain to me about morphology and fitness as you have been thinking about it in your group. Jackson.
So kind of what we were going with was longer legs. So longer morphology would lead to increased sprint speed.
So if you have long legs versus short legs, you might expect speed to covary from slow speed to fast speed. Why would you expect this to happen? Somebody else from the water bug group.
Like sprinters and runners and humans, long legs usually leads to better speed.
Yeah, sure. So oftentimes you see distance runners that are kind of better at endurance. They'll be kind of tall and thin. You generally see sprinters that are shorter but then also pack more muscle on their frame. But if you can pack a bunch of muscle on your frame and simultaneously be really, really tall, like who? Usain Bolt. What was his height again? 6'3", and the guy's built like a tank. You combine that height with all that muscle mass in those legs, and what does he do?
Sets a bunch of records.
He smokes people. He's like
6'5".
Yeah.
6'5". 6'5".
No matter.
So leg length, this morphological character might be correlated with sprint speed for perfectly good reasons. And so because organisms are made up of more than just one trait, you can think of evolution not as something that shapes one particular trait, selection shaping one particular trait, but selection can shape suites of traits. If it's important to be fast, like Usain Bolt fast, and we were in a natural arena where being really, really fast keeps you from getting tracked down by a cheetah and killed and eaten, you might expect Usain Bolt's genes, Usain Bolt's combinations of really long legs and the ability to pack a lot of muscle onto that long-legged frame, those would be genes that would be passed on to the next generation and spread through the population because that's what makes you fast. But it isn't speed alone that is being exerted, that is being subject to selection. Speed, because it's correlated with leg length, is also going to be selected against, selected for. And so in this case, we have two traits that are undergoing similar levels of selection because they happen to be correlated with one another. This is what happens in Thamnophis organoides. So remember in the Brody chapter that we talked about on Monday, he was actually in Oregon looking for snakes because he was interested in how the morphology of these snakes was related to their behavior. And he found over time that striped snakes and checkered snakes behaved differently. And he was curious as to why these two sets of behaviors, these two sets of traits rather, behavior and morphology, would have come to co-evolve with one another, such that striped snakes tended to run in a straight line, and checkered snakes tended to reverse their direction fairly often. And so this required him to think about quantitative genetics in a multivariate way. Now we can think of stripedness and reversals as univariate traits, so we could actually look at stripedness. Let's say that the heritability of stripedness is 0.707. And so if this is the average stripedness of the parent, and this is the average stripedness of the offspring, if there's positive selection for stripedness, making snakes stripier, you can quantify that as the difference between the parental population before and after selection. And if the trait is heritable, that will lead to a change in the offspring character that would have been there before selection versus the one that is produced after the parents have undergone this selective event. So if it's important to be stripy to avoid predators, you should expect selection to push towards more striped snakes, and that will cause the offspring population to increase in its stripedness. You see here that we're, I don't know, at about 13 or something like that, and we go up to maybe 16, something like that. In this case, the average stripedness before selection was 13, the average stripedness after selection was 15. That's the strength of selection. Selection is always the post-selection phenotype minus the pre-selection phenotype. The reason for this is if selection acts to make a trait larger in phenotypic value, the post-selection will always be bigger than the pre-selection, and this whole thing will be positive. If selection acts to reduce the phenotypic value of something, the post-selection value will always be smaller than the pre-selection value, and this will end up being a negative thing. So negative selection causes phenotypic value to go down over time. Positive selection causes phenotypic value to go up over time. And so that's the reason that we always calculate selection differential here as post-selection mean minus pre-selection mean. We take that difference, we multiply it times the heritability value, and then we can calculate how much we expect the next generation to change in terms of phenotypic value. Where do we get the heritability? Where do we find heritability here? What is it? 0.707, because that's what you multiply times the parent phenotype. We can also do this by basically looking at the equation and say, well, if the parent phenotype, the average parent phenotype after selection is 15.42, translating that through selection and the y-intercept will give us the post-selection value of the offspring phenotype, figure out what the average offspring phenotype would be at an average of 13.13, and then subtract the post-selection minus the pre-selection average of offspring. That will also give you the response to selection. There are a couple of ways that you could think about this. Probably the easiest one is to use the Breeder's equation. So remember that this equation is called the Breeder's equation because people can use this to predict, for example, how do I need to select my cow herd so that I get better dairy production, I get more quarts of milk per cow. And so the Breeder's equation basically is this equation where we have the response to selection is the heritability times the strength of selection. Did you have a question?
Yeah. For finding the strength of selection, you said you take the post-selection minus the pre-selection.
No, yeah, post-selection minus pre-selection.
How do you find those values?
You collect the snakes at one period in time, and then you let them go out onto the landscape, and then you collect them at a later time, and you measure the same characteristics on those snakes, and anybody that you can't find, you assume that they died, and then you have a difference there. So remember, when he was talking in the chapter that you read, he was talking about how while he was doing this, he would basically arrive there in the spring when the snakes are coming out. he would race them down racetracks, and he would measure their color pattern, and then he would let them loose, and then at the end of that season, in the fall, when they came back to their hibernacula, he would collect them again, note who came back, and he did that for a couple of years. And so you just look at what the pool was when they were babies, you let them out onto the landscape, whoever comes back, you see who it is, and you know that you presume that anybody who didn't come back probably died. Now, that's not foolproof because maybe individuals don't come back because they went over the mountain or are now in the next valley over, but because he's working with snakes that come back to the same hibernacula typically, collecting snakes at a hibernaculum is actually pretty foolproof. A hibernaculum is a place where snakes go to overwinter. My front porch is a hibernaculum. When you come to my house in the spring, we used to have a little shrub that sat right next to the porch. And in the spring, when it starts to warm up, you can exit the door, and sometimes there will be 5 or 10 little snake heads poking up out of the bush looking around. So they go and they hang out in the bush, and then when it gets cold at night, they go back underneath the porch, and then it warms up during the day, and they come out and they turn their egg, they warm themselves up, and then when it cools down at night, they go back underneath the porch until finally temperatures are warm enough that they just disperse away from the house. But then, after they go out into the neighborhood and live out their life over the course of the summer, they come back in the fall and they crawl out underneath my porch and spend winter there. And it scares the crap out of my mother-in-law when she comes to visit sometimes, which is just an added benefit. So because they have this fidelity to a hibernaculum, they come back to the hibernaculum that they came from. It's usually a pretty good bet that if somebody didn't come back, it's probably because they got killed somewhere along the way. And so you just look at, here was the pool of snakes that we had before, this is the pool of snakes that returned. What's the overall phenotype of that pool of snakes that we let go? And what's the overall phenotype of that pool of snakes that came back to us? Any differences between that represents selection. Now, it doesn't mean that just because you have fewer snakes coming back that selection happened. You may get a bunch of snakes that come back, but on average, they have the same phenotype as the snakes that left, in which case there is no selection. So yeah, it's time consuming, but you can do it. This wasn't really done very often until 1983. Well, it was done a couple of years before 1983, but a guy named Steve Arnold, who was Butch Brody's major professor for his dissertation at the University of Chicago, he was at a party with Russ Landy and some other people. Russ Landy is another quantitative geneticist, super smart, theoretical thinker. And they were basically hanging out at the Great Lakes at the Indiana Sand Dunes at a party. And according to Steve, if you ever get a chance to talk to him and ask him about the story, which if you ever meet him, you should, according to him, they were basically pretty hammered and went down to the beach to just kind of watch the waves lap against the beach. And while they were there, they noticed that a bunch of water bugs were washing up onto the shore. And they noticed as they were washing up on the shore in their states, they were still biologists, hardcore biologists, they noticed that some of the bugs that were washing up on the shore were clearly dead, and some of the water bugs were still alive. And they had seen earlier in the evening, off in the distance, over the Great Lakes, these thunderstorms. And what they thought had happened was that the storm must have knocked these water bugs that were migrating down into the lake. Some of them drowned, some of them didn't drown. The ones that drowned probably were able to maintain their flight longer than the ones that did drown, and they got washed up onto the shore, and some of them were dead and some of them were not. And so, according to him, they got a bunch of empty beer bottles, which I guess were plentiful at the time, and they ran up and down the beach, picking up bugs and putting them into beer bottles that they kept separate for live bugs and dead bugs, and then took them back to the lab and looked at their wing morphology. And what they did was they looked at the population of all of the bugs, and then they looked at the population of bugs that had died versus those that had lived, and the difference between those that had lived and the whole population of bugs. That was the strength of selection on that particular night at the Indiana Sand Dunes. It's a pretty good story. They got a paper in evolution out of this. It was published in 1983, which was kind of the first modern modern attempt to start putting numbers on these kinds of things. I don't get to trot the Indiana sand dune story out very often. We could do the same thing with reversals. So we did it before with stripedness. We can do it with reversals. What's heritability of this trait compared to the last trait?
Much lower.
It's much lower, isn't it?
About a third from last year.
Yeah, so the slope of this line is lower. So if selection acts on this trait, even if the strength of selection is higher, what do you expect the response of selection to be? The response to selection, you expect that to be lower because the heritability is lower. We can do the same equation. What was selection? Whoa, let's go back. Oh, selection is acting in the opposite direction here. So there's selection against reversals. I'm like, why are those numbers messed up? As soon as I flip to the next one, I went, wait, these numbers are wrong, but they're not wrong because the direction of selection is it's serving to decrease the number of reversals. So if there's selection against reversals, you would find that before selection it was 1.94, after selection it's 1.35, so this is selection that is negative directional selection, not positive directional selection. And so what you should expect is you should expect the phenotype reversals to go down over time. Once again, you can do this with the Breeder's equation, but you can also do this other ways that are more complicated. So what the Breeder's equation does is it allows us to look at the heritability of a trait, and if we know something about the selection of a trait, we know what that trait is going to do in the future. If you're a milk producer and you know the heritability of milk production in cows, and you want to increase your milk production 20%, you can then calculate how strong the selection needs to be on your milk herd in order to achieve that level of increase in milk production. And so it doesn't matter whether you know heritability and selection and you want to predict what the response to selection will be, or you know what heritability is, you have a desired response to selection, that then tells you how much selection you need to exert on your domesticated animal to get them to achieve the milk production that you want, which is, once again, why it's called the Breeder's equation. This works primarily for directional selection, because remember that these things are moving either in one direction or another direction. There's another form of selection called quadratic selection that looks at shape, so it can detect stabilizing selection, but for now we're going to keep things simple and just think about the phenotypic distribution shifting one direction or another direction. This type of selection that is up here, which is directional. But don't forget that there's also stabilizing, where it just makes the distribution narrower over time, and then disruptive, which we talked about a lot during the Hendry et al. Paper, because presumably that's the type of selection that was driving these finch beat distributions apart. Quick review of what we've done in the past. This has all been reviewed so far. Does it feel like a review, or does it feel new? Okay. So let's now think about two traits that might potentially be evolving in tandem with one another. So Thamnophis organoides is a polymorphic snake. What do we mean when we say something is polymorphic?
Multiple genes,
It has multiple what?
or multiple alleles that are shown.
Okay, so that's traits can be polymorphic, where you have multiple alleles. But in this case, we're thinking of overall morphology. What does polymorphic mean?
One, like, have multiple forms of morphology.
Yeah, multiple forms of morphology. In this case, in the same population, actually in the same little valley at 10 Mile Creek, you pick up a snake. It's in the species Thamnophis organoides. It could be really stripy, it could be really checkery, or it could be anything in between morphologically. And so one of those questions was, how can all these different color patterns coexist together in this same population? If color pattern is important, you would expect, if color pattern is important for survival, you would expect the population to converge on a single color pattern. In the same way that when you look at monarchs, for those of you in the butterfly group, do all monarchs look virtually the same?
Yeah.
Yeah, presumably because that works for them.
Yeah.
And so one of the questions when we looked at this population was, why are there so many different color patterns present? He developed a way that he could assign a quantitative value to how stripy you are, and I to this day don't know what that method was, so that he could measure stripe on snakes. And then he also, with his father, this is Butch Brody Jr., raced these snakes down a racetrack similar to the racetrack that you guys didn't use for your water bed experiment, but the one that I used for gecko racing. And he had morphology and he had performance, and he was interested in whether or not these two things were related to one another. So, in the same way that you can calculate heritability by looking at trait 1 in parents and offspring, and now I've erased the heritability figure up here, you can also look at genetic correlations or genetic covariances, but in this case, what would we have on the y-axis if parental stripedness is on the x-axis?
Offspring reversalness?
Well, so stripedness goes from a high value, which means pretty stripy, to a low value that means pretty checkered. And so you would have reversals. You would have offspring reversals. Average offspring reversals. How much of a tendency do they have to reverse themselves from a low tendency to reverse themselves to a high tendency to reverse themselves? And this can take on more potential values than heritability can, because it may be that there is no relationship between stripedness in parents and reversals in offspring. It could be that there is a positive relationship such that parents that are more stripy tend to reverse themselves, tend to have offspring that reverse themselves more. Or you could also have a negative relationship where parents that are stripy tend to reverse themselves less. This is a genetic covariance of 0. This is a positive genetic covariance, and this is a negative genetic covariance. But it's genetic because it's looking at offspring and parents. That's how you get the genetics, by looking at it across generations. We could do this just within the parents, but that would be a phenotypic covariance or correlation, not a genetic correlation or covariance. It's only when you do this across traits. So when selection acts on, if there is a genetic covariance, when selection acts on one trait, it also drags the other trait along with it. And that's why this might be important. Everybody okay with that as an idea? So I did this on your PowerPoint so that you have it. Actually, I got the order wrong, stripedness and reversals. And in the case of this particular situation, it could have been a positive correlation, but it turns out that in these snakes, there's actually a negative genetic covariance or genetic correlation between these two things. Striped snakes tend to reverse themselves less often. Checkered snakes tend to reverse themselves more often. And so there's a negative genetic correlation, not a positive genetic correlation, when you go and measure it with these parent-offspring regressions. So if you can model the evolutionary response to selection with one trait, you can model the evolutionary response to selection of as many traits as you can think of. But you're going to have to do this with a new kind of math. Before we had heritability and selection affecting the evolutionary response to selection, now we're going to have a matrix of genetic variances and covariances, and we're going to have a vector of selection gradients. And so just don't be frightened. I'm about to show you something that might make you freak out a little bit, but relax. We're going to work through it, so just don't freak out. This is what it looks like. But all this is G11 is the genetic variance. It's kind of thinking about the correlation between trait 1 and trait 1. And so when you think of these ways of measuring heritability, when you measure heritability, you measure mid-parent value for trait 1, and you measure average offspring phenotype for trait 1. So this first G here is the heritability, essentially, or the additive genetic variance, if you want to call it that, of trait 1. What is G22?
The genetic variability of trait 2.
The additive genetic variance or heritability of trait 2. What is G1 and 1,2 and G21? It's the correlation between trait 1 and trait 2. Reversals versus strikedness. The degree to which two traits are genetically covariating with one another.
Did you say what G11 and G22 are again?
It's the heritability of trait 1 because you're looking at the offspring phenotype of trait 1 and the parental phenotype of trait 1. All you have to do to get the genetic covariance is regress trait 2 against trait 1. And so in this matrix, you have not only what the heritabilities of the individual traits are, but you also have how they're correlated with one another or not. And it could be that they're not correlated, in which case these two things are 0. It could be that they're positively correlated, in which case these would be positive, or they could be negatively correlated, in which case these would be negative. These are always going to be positive. The question is, are they going to be positive and close to 1? Are they going to be positive and close to 5? Are they going to be positive and close to 0? Does that make sense? Jonas,
Yeah.
question?
I just realized what was happening. I'm bad at matrices, and so my brain is ticking through all of this.
We'll work on that in a few minutes.
And yeah,
Beta is selection gradients, which is different from a selection differential. Yep. Somebody say something?
No, I just want to make sure everyone can break down the term genetic covariance, right? The genetic portion means you're regressing to the offspring and the parents. So you get that generation, that's your genetic portion. And the covariance of two separate traits. So if you need to dissect that term out, you got to know there's two components. And that's what G21 and G1 are.
Yes. And those of you in the Waterbuck group, when you ran a correlation between, for example, body length and femur length, you were basically measuring the phenotypic covariance of those two traits with one another. It's not genetic because you didn't have parents and offspring. You just had those two traits within an individual. And so covariance is just used anytime we have two traits that can vary in concert with one another. That's the covariance. Beta is a selection gradient. And S is a selection differential. And this is a kind of important thing. When you do, the responsive selection is due to heritability times selection. Because you're only looking at one trait, you kind of make the assumption that this trait is the only thing that really is important. So, if I'm looking at a trait, what would be a good example, Dr. John, of a trait that might be correlated with another trait?
Well, the ETE group that I was with, California, almost all
Okay, I'll just run with that. So oftentimes in organisms, what you see when you look at body size is body size from small bodies to large bodies is often correlated with reproductive output, how many offspring you have. And so if you wanted to look at the evolution of body size, you could go out and you could measure the heritability of body size. Do large-bodied parents give rise to large-bodied offspring? You would find the heritability of body size. You could look at differences in selection in body size and find what the selection is, and you can predict the evolutionary response to selection of body size. But in this case, body size might be evolving because what's really under selection is reproductive output. How many offspring do you have? In which case, body size isn't the object of selection, but it looks like body size is the object of selection because body size and reproductive output are correlated, and reproductive output is actually the thing that is being acted on by selection. But when you only have one trait that you're looking at, you don't know that. So a selection differential includes direct selection acting on the trait you're measuring. Plus any indirect selection directed at other things. Maybe there's no selection acting on body size at all. All the selection is being acted on reproductive output, and body size is being just drug along for the ride.
A lot of mammals have uteruses with folds in them, and that controls litter size. So you could maybe have selection for litter size that would be related to reproductive output at a given body size.
So when you're doing this now in a multivariate way, you want the selection that you use to not be selection that could be indirectly influenced by other traits. And so what you do is you measure selection in a way that makes it independent of other traits. And so what you do is you look at fitness as your Y variable, and you basically do a multiple regression in this case. So those of you in the water bug group, we did a multiple regression where we looked at each individual leg segment and looked at how those are correlated with speed. When you do that, so fitness in this case, you might have a y-intercept in the model plus, but then you would have a correlation coefficient that is due to reversals. You would have multiplied times the phenotypic value of reversals, plus the correlation coefficient for, in this case, stripedness, multiplied times the value of stripedness, plus residual unexplained error. And these correlation coefficients are what they call partial correlation coefficients. They're the correlation between this trait and fitness holding this trait constant. This is the correlation between stripe and fitness holding reversals constant. And in this way, you can statistically remove the correlation between the two traits in assessing how this is related to fitness. And the relationship of this to fitness is exactly what selection is. And so this gradient gives you how is the selection for reversals acting on reversals independent of any selection acting on stripe. How is selection acting on stripe independent of any selection acting on reversals? And that becomes important because when you want to know how reversals might be evolving, you take the heritability of reversals, you multiply it times selection that only acts on reversals, and then you add that to the genetic covariance between stripe and reversal multiplied times selection acting on stripe independent of reversals. And then that tells you how reversals changes over time, but it'll also tells you the degree to which that is responsible, the things responsible for that is selection acting directly on reversals versus selection acting on stripe and affecting reversals through this genetic covariance. We've already been through this. So we can rewrite this genetic covariance matrix to show that this is the additive genetic variance of reversals, this is the genetic covariance between reversals and stripe, and this is the additive genetic variance of stripe. Heritabilities in the upper left and lower right, and genetic correlations or covariances in the upper right and lower left. And we measure this by parent-offspring regressions of one trait. Oh, sorry, this is heritability of reversals because the same trait is on the y-axis as is on the x-axis. What is this? So it's a genetic variance or a genetic covariance? Genetic variance, good. This is
The genetic code.
This is genetic covariance, and this is the genetic covariance that he observed, where individuals that tend to reverse themselves tend to be more checkered, individuals that don't reverse themselves tend to be more stripy. I've already given you what the selection gradient is. It's the directional selection acting on that trait independent of any selection acting on another trait. It's basically this. Such that if this is the heritability of the reversals, it's selection acting directly on reversals without taking into account that reversals and stripe are correlated with one another. Same thing with stripe. And then if you know those two things, you can predict the response to selection. So how does matrix algebra work? Well, I will help you with this. I find the easiest way to do this is with a pen that works. This is what it looks all worked out. Let's go ahead a couple of things. Wait a minute, did I do something wrong that I wanted to not do? Oh yeah, so think of this as trait 1, trait 2, trait 2. So basically, if we want to know what R1 is, what the evolutionary response to selection is, we take G11, we multiply it times beta 1, we take G12, multiply it times beta 2, and add those two things together. Then we take G21, multiply it times beta 1 for R2, we take G21 times beta 1, because this is the effect of selection on trait 1 translating into change in trait 2. And then we take G22 and multiply it times beta 2. You always want to, whether you're doing trait 1 or trait 2, you know where to start because you're predicting what happens with trait 1. So you want the heritability of trait 1, so that's going to be 1, 1, and you want selection on 1. That tells you where to begin. An easier way to do this one down here would be to say, well, I'm looking for the evolutionary response to selection on trait 2. I need to start with G22. Well, what am I going to multiply that times? I want to multiply that times the direct effect of selection on trait 2, beta 2. Well, if I'm going to do that, then I want to know the indirect effect of selection of trait 1 on trait 2, beta 1 times G21. And those things added up will tell you what direction selection is going. Yeah.
Okay, I was kind of following along with PowerPoint, because on here it says G21 times B1 for the second trait.
Yeah. What did I say?
G22.
Oh, well, I just switched them.
Okay, so it's the covariance first.
This is G21 times beta 1. And what do I have? G21 times beta 1.
Yeah, you just reversed the order.
I just changed the order.
Okay.
If you want to kind of keep yourself straight, you can always know that if you're dealing with trait 1, this one goes with this one. If you're dealing with trait 2, these two 2's go with this 2.
Okay.
Okay, does that help?
Yes.
So I'm trying to systematize things for you. All right. I think we need to hand out exams. So
This is homework?
yeah, we'll do this as homework. Have homework. We were going to do it in class, but I'll just have you do it as homework, and then come back to class on Friday with this done. Dr. Judd will hand out your exams. Your performance on the exam was not happy telling, unfortunately. You missed some things that we really thought you wouldn't miss.
So we found that in general, there were pretty consistent places where you guys got your points and where there were issues. And I think the point is pulling this up. This was one.
So the activity that you passed on to homework.
Yeah, do it before class on Monday. This is the distribution. This is not the kind of distribution that we were expecting. Surprisingly, a lot of you missed the last question on the exam, which had to do with the carbon cycle. Dr. Judd and I could have passed these out on Monday, but we wanted to talk about this. I went back and looked at the video. We spent 20 minutes in class on the last question on the test. So, and then when we got finished with nutrient cycling, Jonas asked a question that made me go back and revisit that. And I told you, once again, you can go and see this on the video if you want. You don't have to trust me. But I said something along the lines, he was asking about budgets and whether the nitrogen budget was balanced or unbalanced. And I said, well, it's a little tricky because there's a lot of variation in nitrogen in soils. But generally, we think of nitrogen as being unbalanced because a lot of nitrogen leaves ecosystems in stream flow. And then I said, but don't worry about working a budget like we worked the budget on the carbon cycle, because I want you to know the carbon cycle in much more detail from a budgetary standpoint than the nitrogen cycle, simply because it's easier. It's a little less complicated to think about. And so you were also clued in by an interaction that occurred later on in the class. So when we got to the test, and like 10/12 of you missed virtually all of the points on that. And I think it was like 10/12 or 9/12, something like that.
Yeah.
So the vast majority of you missed virtually all of the points on that question. We were surprised because we did spend quite a lot of time in class on that particular thing. So then we're like, okay, well, what do we do about this? The grade distribution isn't really very happy looking, but we discussed it and we feel like you kind of had ample opportunity to kind of get the point of that. So what we're going to do is on the final, we're just going to tell you that exact question word for word is going to be on the final. We're not going to tell you the exact nature of all of the other questions that are going to be on the final, but that question will be on the final in its current form. Because it's a fundamental kind of idea to think about a nutrient cycle being budgetable, determining whether things are being stored in ecosystems or being exported back to their main pools. You can think about that with reference to phosphorus. You can think about it in reference to nitrogen. So it's a pretty important idea, and either I didn't do a very good job of communicating it to you, or you didn't do a very good job of studying for it. Is that?
So that's why we are putting it on the final.
That's why we're putting it on the final. We want you to know it. We think you should know it.
If you want to go over the other areas that were sort of hot points, were the ANOVA table and the Tillman style diagram stuff. So if you want to go over any of those things, we're happy to talk to you